We travel around the world and we actually try and collect different interesting stories that are happening globally. And well, you've got quite an interesting story happening here at Jefferson. Want to tell me about it a little bit?
Certainly. So as you know, I direct the Computational Medicine Center. We are fairly new as an organization. We were founded, I'm the founding director, in 2010. And we focus on human disease, but we have a different view on human disease, one that's informed by work that we have been doing in the center.
The different view, talk about that. I mean, because it truly, there's a lot there. I mean, you're taking on the Goliath of cancer, but you're also taking on way that research is actually practiced, way medicine is even practiced, and you're looking at a lot more information than ever has been done before. Maybe you could expand on that a little bit.
Excellent point. Starting from what you just said, the thing we're doing differently is we take a view of the human cell, which is non-reductionist. Reductionism helps, as you know very well, in the sense that it makes the problem easier to deal with. It has benefits, but also it has limitations as an approach. And what we are saying is, let's let the data tell us what's important in a cell. And this, in many ways, is the opposite of reductionism, because as we're discovering, the data is telling us that there are more variables, more things to consider, and this is precisely what we do. We take all of these new variables into account as we try to understand the onset and progression of disease.
So reductionism it's a function of, like, as an example, I could walk in and say, I've got a head cold, my nose is running, my daughter's also sick as well. And reductionism would be, well, you would kind of eliminate, it's probably not environmental, it sounds like it could be viral, and then you reduce it to it might be viral and you chase that. You, from a research perspective, like, see it very differently. You're more inclusive, meaning include more data. And if you think about it, it's kind of basic thought. It's what we all learned in statistics. Like, the more data that you look at, the more accuracy that's actually driven out of the work, and that's sort of your approach.
Yes, it's true. The more data, the more accurate you are. The limitation that we experienced as a field is that we didn't have data until very recently. We knew that it would help, but we didn't have the technology that would allow us to generate it from contexts of interest. The thing we also do differently is we look at disease bottom-up as opposed to top-down. A practitioner, a clinician, a doctor looks at you, looks at me and says, oh, you have a cold. But that's looking at the problem from the outside in. We come in from the inside out. We look at the cell and we say, okay, this is driving everything that we observe macroscopically. What is it that is actually manifesting itself as a disease? And we try to understand this by first figuring out how many players, if you wish, exist in a cell, who is driving the cell's behavior, under what conditions, et cetera, et cetera. The environment is always a consideration because the environment will affect a cell's behavior.
Disease. What causes some people to develop disease and others not to?
That's a very...
it's a bit broad, I know.
It's broad. It's one of those easy to pose questions that have very long answers.
I'm quite good at that, by the way.
Originally, when we try to understand disease, we, again, had to resort to a reductionist approach. And the molecules that seemed to make sense to pursue first were proteins. And for many decades, we worked on figuring out which proteins cause what disease. And we found them and we created long lists over the years.
Kind of the opposite of reduction, right? Now you're creating long lists and looking at lots of information. You're completely coming at the problem of disease in a completely different way.
True, nonetheless, you're still looking at a finite set called proteins. And in the early 2000, before the Human Genome Project was released to the public, people actually placed bets as to how many proteins will we find in the human genome, how many protein coding regions will we find in the human genome. And looking back at those numbers, you can actually see that there were very wide differences in those numbers.
Were they close? Was the bet right? Or
no, not really. Many people thought the number was much higher than it is.
In what range?
I seem to remember some groups were proposing as many as 30, 40,000 proteins. Others went even higher, more than 100,000. You have to understand that this is informed by our observation that humans are complex.
Sure.
So if we try to explain everything by reducing it to the proteins that are driving this apparent complexity, then you would expect a large number, or else you cannot explain the complexity, right? So it was, for the high numbers, it was expected because we had to explain we had to explain us. On the other hand, we know it's a very complex system, and you can have complexity with a few parts. And some people were voting for the first approach, others were voting for the second, some were in between. When the genome was released, we realized that the number is about 20,000.
20,000?
Yes.
Versus 100, 300,000. That's quite a miss.
Correct.
Call that not a good bet, right?
Yes and no, because it came with a twist. 20,000 was the number of the locations in the human genome that made proteins, and then people realized that some of these locations would make more than one proteins. Gotcha. With slightly different properties. We still don't have a good grasp on how many proteins are ever made in a given cell. The last number that I saw, it's probably 2 years ago, was in the high 50,000 estimated proteins, distinct proteins, made out of the 20,000 loci that are currently accepted to be protein coding.
Was that sufficient to answer the disease question?
In some respect, yes, in the sense that we started seeing that proteins and disease are linked, and we were able to find instances where changes in a protein coding region manifested as a disease in a person that had these changes. So we were able to link changes in somebody's DNA to disease. Where it started getting interesting and perhaps more complicated for researchers was there were instances where we had disease, but there were no apparent changes, at least in the places where we looked, namely the protein coding regions.
So you have disease, yet you don't seem to have changes in the protein. What's going on there?
It was a puzzle for a while, and it so happens that here at Jefferson in the year 2004, there was a first discovery that actually led us down a path that was very promising, and it has proven tremendously useful. In early 1990s, we discovered a group of regulators called microRNAs. So these are very short molecules, but very powerful. And what they do is they regulate the production of proteins. If they themselves are dysregulated, then you can see that the levels of protein may be more than you need or less than you need. So you have a disruption of the system. What was unclear for many years was whether that disruption can cause disease or not. And what was shown in 2004 by former colleagues of mine here at Jefferson was that indeed the absence of one of these regulators can cause disease. That was the first instance where we actually linked regulators to disease, even though the affected proteins were not changing. They were still present, but present at quantities that were not the quantities you encounter in a normal cell.
So, I mean, 2004, that's 14 years ago. You have this aha moment at Jefferson. What runs through your mind at that point? Are you like, we're on to something, I see it? Because the day I spent here with your staff and team, like, it was just a tremendous amount of pride, this passion that they're moving towards a goal, right? And you feel that. Back 2004, you must have been elated or just, wow. I mean, what's the feeling that you go through from then to now, and where do you see it going?
Interestingly enough, in 2004, I wasn't as immersed as I am now in this area. I was working on microRNAs myself. Mm-hmm. I was trying to solve a different problem, which is if you know the sequence of a microRNA, can you tell me which proteins it regulates? It was an important thing as a problem. We knew microRNAs are important molecules. We hadn't linked them to something of true importance, such as a disease. At the time, I couldn't appreciate the magnitude of what had been uncovered with this first discovery.
So you know, but you know it now, right?
Oh, absolutely.
Back at the time,
looking back,
it's
absolutely
not as apparent as it is today.
To the people who made the discovery, it was very clear. But they had been working in this field for much longer than I had. I started working on microRNAs in 2002. And I was still focused on perhaps a very limited picture of what microRNAs can do, which is, I have a microRNA that regulates some proteins, let me figure out what's being regulated. They were asking a different question, a subsequent question, which is, okay, and how does this matter? Why does this matter? So they were answering the next question. Eventually, obviously, I realized that this was an equally important, if perhaps more important question. And I have been working on disease-related questions pertaining to microRNAs ever since.
So my question clearly wasn't that simple. So this discovery of microRNAs, is that it, or is there more?
For better or for worse, there is more, as is always the case with research and science. So, as we discussed, we collectively as a community linked microRNAs to disease. And then we started studying how different microRNAs are relevant for different diseases. So for every disease, microRNAs control some of the proteins that are relevant for that disease. The assumption we made as a community was that all of us had the same microRNAs regulating the same proteins. And what we discovered in my center is that this is not the case. It goes back to something we discussed earlier. We have regions in the DNA that make molecules. Originally, these regions that we cared about made proteins. And we said also that there are 20,000 regions, and we thought they make 20,000 proteins, but we learned that they make more than that. We just don't know how many more. The same thing happens with microRNAs. The region of the DNA that describes a microRNA, for many years we thought makes one regulator, one microRNA.
Right.
And what people saw when the technology permitted us to do that was that multiple molecules were coming out of the same region. Originally, the immediate reaction was, oh, these other molecules are probably not important. They are noise.
So back to the reduction thinking.
Yeah.
You know, pervasive, clearly,
yes, I mean,
in the industry.
All of us are trained to be reductionists. That's what we learn in school.
I mean, across the board. So,
Absolutely.
I mean, it's not just research,
Absolutely.
not just science. I mean, care, treatment, medicine.
Absolutely.
Reduce.
And there is a good reason for that. It makes the problem more manageable.
Absolutely. It's how a mechanic approaches a car engine. It's how a plumber approaches a plumbing problem. No different. It makes a lot of sense.
Exactly. And what we discovered was, well, actually what we suspected was that these other molecules may be there for a reason. Originally, we didn't have enough data to answer that question. Eventually, the data became available. So we had our first publication in 2014 where we actually showed that the variance of a given molecule that you observe being produced from the same region of DNA differ based on variables that we never considered.
Like what types of variables?
For example, a person's gender.
Sure.
We showed that men made different variants than women. And not only did the variants differ in terms of identity, even for variants that were the same, the relative abundances changed as a function of gender. We also showed that a person's race impacts on which variants will be made and how much of each variant.
And that makes sense because you've seen that in disease. You've seen different racial impacts of disease associations.
Very true.
And certainly with men and women, so
Very
makes
true.
sense.
It makes sense, but remember when we make that statement, when we make that observation, we're looking at the problem from the top down.
Right.
In other words, we could observe it, but we had not had the luxury of finding the mechanistic events that may underlie this.
Are there other variables? So you've got men, women, race.
Yes.
What other variables?
You can have people of the same race that come from different geographies. We showed that this matters as well. They make different combinations of variants as well. We also showed that a person, you can define a person in terms of gender, population, and race, and the only thing now you do is you look at different tissues in this person. We showed that the same piece of DNA will make different variants out of the same microRNA region as you go from tissue X to tissue Y. Keep in mind, these molecules are regulators. They regulate protein abundance. And we already established that changing protein abundance can lead to disease in the absence of mutations.
I have to imagine there are other variables too, like lifestyle, foods you eat, smoking, not smoking. Are those factors as well, or?
People suspect them to be factors, but there isn't enough data yet to actually connect the dots.
Right.
But if I had to guess, I would say the dots will be connected once the data is available.
Well, you're clearly like radically moving away from reduction to. I mean, you're adding yet more and more variables, and you're proving that it has an effect. It truly does make a difference.
Correct. The problem you have, though, as a scientist who tries to understand something supposedly simple as a cell, is that what you thought is manageable has become less manageable because now you have all these things that come into play. And again, going back to what you said, looking at the person from top down, from the outside in, you know that somebody who exercises typically lives longer than somebody who doesn't. What we didn't know is how that external event, exercise, or the food they eat, manifests itself or translates into events that happen inside the cell. Everybody thinks there is an actual connection.
It seems logical.
It seems logical.
But yet still unproven.
Just because we don't have enough data, people are beginning to realize that all of these connections are there and they are generating data to demonstrate, to connect the dots, essentially.
Does the data, I mean, curious, does the data exist or has it just never been connected?
Some of it. Some
there.
Yeah, some is there, and it looks very promising. There are data that basically show that somebody's diet affects the abundance of microRNAs that you can have. And this was shown both in non-human primates that were put on a diet, as well as in cells from human tissues.
So, wow, quite a question, quite a problem, by the way. So are we done, or are there more?
Unfortunately, yes.
Yes. Fortunately, yes.
Yes. It keeps us busy. It's exciting to discover new things, but in terms of the problem being manageable, it's even less so now. While all of this was going on, while we were reporting that variations of microRNAs were affected by variables like a person's gender or race, some other groups were reporting another category of short molecules called tRNA fragments. They were discovered in deep sequencing data, meaning in data that became available to us as scientists because of a technology that emerged in around the year 2006.
Right. So you have this 2006. You have this aha moment in 2002, and in 2006, deep sequencing becomes apparent, and you're presented with more data. And now what?
In 2006, the technology emerged, but it was still very expensive, and eventually the prices went down, which made the technology available to more people.
It's come down quite a bit, though. Correct? It was prior. It was few people could do it, and now almost everybody can. It's affordable, for lack of a better word.
Absolutely, and in fact, it's a blessing for us, because that means people can generate data from their respective context, the problem they've been studying. Eventually, the data becomes available in the public domain, and people like me, who try to connect dots across different contexts, all of a sudden have all the data we need to make these connections. So what happened with the tRNA fragments is very similar to what we discussed with microRNAs. So the deep sequencing data showed us that the molecules called transfer RNAs, or tRNAs, which were discovered more than 60 years ago and are important for translating a messenger RNA into protein, in addition to having this long molecule, you were seeing fragments of that. And again, our reductionist upbringing essentially said, oh, these are probably noise.
I suspect they're not, though, are they?
They're not. And again, my group was first to show, just like we did with the microRNA variants, that these fragments depend on variables that we never considered.
So your group again,
My group again, yes.
So as I said, as we started the story, it's quite a story here.
The problem is that we are taught to look at numbers and draw conclusions. Personally, it's
your background, right?
My
You've got a long background in that.
But until you actually show that molecule X is responsible for event Y, it's very difficult to convince people. Going back to what we said before about 2004, until it was shown that a microRNA is linked to a disease, people were questioning the magnitude of what microRNAs are able to do. There was always a question mark at the back of people's minds, and that event sort of changed everything in that regard. tRNA fragments, there was a similar reaction initially, namely that, oh, it's probably noise. So we are showing that they are controlled, how much of a given fragment you'll make and how many fragments you'll make. Again, as we showed, the function of a person's gender, a person's race, the geographic origin of that population, the tissue, whether it's the tissue is in disease subtype X or disease subtype Y. Now, having said that, the immediate question that everybody asks is, okay, so I'll show you this molecule, tell me what it does. And this is where we stop currently because we haven't answered that question. The practical problem is the number of distinct molecules that fall in the category that we just discussed, the tRNA fragments, are so many. As of last count, we reported as a group in late December of 2017, 26,000 distinct tRNA fragments across almost 12,000 people.
Wow. It's quite a number considering what you thought it was originally, that the community thought it was.
Correct. So not only do you have a new category of potential regulators, and there is enough evidence already to believe that tRNA fragments are regulators, just like microRNAs. So originally, remember, we started with proteins. We thought that's it. Then we discovered microRNAs. Then we discovered that microRNAs come in variants that depend on other things. Just the variants alone increased the number of molecules in the microRNA domain by a factor of 10. So we know there is 10 times as many variants as the places on the genome that make them. Wow. And these variants depend on the patient characteristics. Now, that's one category. Then we add a second category of molecules. We suspect all of them or most of them are regulators, and we already have almost a little over 26,000 of these new molecules. And they are regulating the things we care about, such as proteins. In the context of disease, trying to understand what all of these molecules do to the proteins that we all agree are behind diseases, it's a Herculean task, right?
Great word for it. Yeah, truly, right. Truly.
And based on what we have seen, just understanding one microRNA molecule can take a graduate student maybe 5 years, and they'll understand it only in a very specific domain, namely a tissue, for one disease. And that same molecule could have a totally different behavior if you go to another tissue and you talk about another disease. And we are discussing tens of thousands of molecules here.
Incredible.
Yeah, it's humbling in many ways.
Certainly is.
We understand that. We understand that we're not going to answer these questions before each one of us retires.
I mean, I guess the million-dollar question would be, how long does it take us to get there? Is it 5 years? Is it 10 years? Is it further?
Excellent question. It's a function of what do we mean by getting there. Where we find ourselves right now is a situation where we discovered all these new molecules, and we do not know what they do. Essentially, it's as if I handed you a dictionary for a language you do not speak, but with one twist. I removed all of the meanings from the dictionary. So essentially, I handed you vocabulary, but you don't know how to use each one of the words.
So you know all the words.
You know the words.
None of the meanings. And your goal, deliver the meaning.
Everyone's goal
for every word.
Correct. Everyone's
that
goal.
That level of magnitude.
And the problem is, I have to first convince my colleagues that it's worth understanding what this vocabulary is all about. That part will probably take 5 to 10 years, in the sense that just like microRNAs before, I suspect somebody, not necessarily my group, but somebody in the community will show this one key experiment where they linked one of these many molecules in the vocabulary to something really important, truly important.
Do you think you convinced them? I mean, do you feel confident that you convinced them?
We saw this already once with the microRNAs, and there was this one experiment that was the turning point that shifted the entire community's attention to these molecules and to understanding these molecules.
So you're not without precedent. It's happened before, exactly. and you're confident you can repeat that.
If not us, necessarily. Somebody in the community will show. We are certainly convinced. And remember also, we have the ability to look at numbers and make a guess as to whether they tell us something important or not. Everything we've seen says they're telling us something important. We haven't, we people in my center have not had the opportunity to show this one key experiment just yet. I know many groups are trying to do that. We are certainly one of the groups, but it's also a function of luck, funding, determination,
Time.
24 hours. Exactly. 24 hours can make or break a result, in the sense that if somebody else publishes it first. But even if they do so, even if we're not the first, it's still useful because somebody actually showed that these molecules are important. And this is the very first step. We need to convince the community that they need to look at these new molecules, the tRNA fragments.
Assuming the community is convinced, what happens?
Remember, we originally attempted to describe disease with the help of proteins, and we achieved that. It gave us knowledge we didn't have. Now we have more molecules that matter. We have the microRNA isoforms and tRNA fragments. For the microRNA isoforms and tRNA fragments, what we need to do is connect the dots. Let's assume that the community connected the dots. What I suspect will happen, and we have preliminary evidence in that regard, is we'll be able to diagnose disease better, and we suspect also earlier. Why am I saying that? Because these regulators are essentially signals of disease onset and disease progression. And we suspect that they don't appear on the scene just abruptly, but they start increasing monotonically their abundance or decreasing monotonically their abundance as an individual progresses toward disease. Knowing that these molecules matter and studying them will allow us to actually capture them in flight as they move through time, which will allow us to create better diagnostics.
That seems significant, though.
We think so. We think so. And also keep in mind that these molecules are, how should I say, are dependent on a patient's individual attributes, gender, race, population.
There we go again. More data, more information.
And those diagnostics now will be fine-tuned for people who are men or women who are from Northern Europe or Southern Europe or from Asia. In other words, you have diagnostics that are extremely powerful because they are designed to address the question for the population of interest.
So to me, I mean, we look at precision medicine quite a bit. There's a lot of focus on it. There's a lot of work there and success there. But this seems more precise. It seems like by you widening the amount of data and the amount of variables and the information, it sounds like you're really being far more precise with precision medicine.
It's more precise precision medicine. The downside of this is it's the opposite of reductionism. So again, from a practical standpoint, you have more things to worry about, more variables that need to be taken into account when you do an experiment. And you can see how this snowballs now into a more difficult setting in which to execute any experiment that you might be interested in.
Right. But, I mean, again, we've learned it in any stat class we ever took, we learned that the greater the amount of data, the more accurate that you are, the more precise you are, the better the medicine. You know, so there's precedent in everything we've ever done with data to prove that you're going to be right.
The only complication with that is when there comes a time to actually practice this, everybody's...
Well, yeah, of course, because you go from, like, let's narrow the problem and let's treat and approach to, like, whoa, wait a minute, we're going to go the other direction, we're going to make it more complicated. and that you have a natural inclination to go, that doesn't feel right, right? So I can imagine you get a lot of resistance to that.
Yes, and in some ways, we are also concerned because knowing that this complexity is there and having shown it's there, we have to account for that complexity ourselves in everything we do, right, which basically forces us to design experiments in far more complicated ways than previously. At the end of the day, it's all valuable because the experiments are designed more appropriately. The results that will come out of these experiments will take longer to produce, but at the same time, they will be more relevant because they are we've drilled down to the very essential events that are taking place.
So, I mean, you can create diagnostics with this new knowledge of diagnostics. Is there anything else that follows?
We think so. Yes. The way to look at these molecules is they are regulating molecules that we already knew are important, the proteins. If they are regulators of important molecules, that makes them important molecules as well. And if you think back, the pharmaceutical industry, our therapies have been focused on affecting proteins because we knew that proteins are misregulated in different disease contexts. Right now, these new molecules are emerging as important parts of this entity called the cell. Immediately, all of these molecules are becoming candidate therapeutic targets. The way to look at it is we have more ways in which we can possibly affect a cell's behavior. We've tried to do that by only focusing our efforts on the subset called proteins. But all of a sudden, we have these other molecules, equally important, that can affect the cell's behavior, and we never examined them from a therapeutic standpoint. So potentially, many of them, not necessarily all, but many of them will become new therapeutic targets.
I'm curious. We haven't talked about this at all, but you've got, I talked earlier. You've got a staff that has a lot of pride and passion. You see it in the time we've spent with them. But you also have graduate students and summer interns coming through the program all the time. So you have this whole thinking of reduce when they come in, and you start to explain your different way of thinking about it. What kind of reactions do you get? I mean, do they resist and just refuse, or do they embrace what you say and become believers, or have that aha moment here?
I think the first reaction is they're puzzled.
So of course they are, because you're basically telling them everything that they've ever been taught might not be the way to think about it, correct?
It's incomplete. We're basically saying that everything we learned is incomplete, which in some ways we expected, because if it was complete, then we would have addressed all of the questions pertaining to disease and therapies. And the fact that we haven't basically says we've been missing on something important. We don't have enough knowledge.
So they're puzzled. And how do they remain there?
For a while, until they get their hands dirty. And of course, they do understand before they join the center. We ask them to read the papers that we published, so they know what they're getting themselves into.
Okay, so they're not. So there's something there that wants them to be here. Absolutely. And then they're here. They're puzzled for a period of time, and then. Yeah. Then what? Then,
while this is happening, we typically expose them into practical questions that they generate data for. So they're working on something that typically they would explain it a certain way. And I ask them to generate data from this different viewpoint. And they have the data, and then they look what they get with the data if you consider the old viewpoint and the data when you consider the viewpoint. Once you actually do it yourself, you realize that you are holding something.
No, it's funny. We actually saw that. We're in your lab, and one of your graduate students. We're filming them to make them a part of this story, and she was so excited to show the work that she was doing, and she was waiting for a particular piece of DNA to become unfrozen. And she goes, "You have to wait 10 more minutes so we can capture this." So we were late to talk to you because she wanted to share that. So I would imagine she showed up puzzled, right? And she's different. I mean, what we captured today is a very different, like I said, passion, pride. Like I want to share this with you. Is that the experience that most the graduate students that come through here?
I should tell you I'm very thankful, and I'm also very fortunate to have these people who work in the center and work with me. And what you describe is true of everyone, from the students all the way to the other faculty in the center. They all have experienced what the implications are of what they're doing. There are several projects going on. We all work together. Everybody is equally excited because they know everything they do will help the rest of the team. And they all understand that we are trying to tackle something which is, on one hand, extremely difficult, on the other hand, extremely important. They fully appreciate that this is a complicated setting, that they'll only add a tiny little contribution to what is evolving into being an extremely complicated picture. But the reality is that picture has always been there for millions of years. And we're beginning to unravel it. It will take time. And they are all committed to beginning this unraveling.
Yeah, they clearly are. It was evident. I've spent a lot of time globally traveling to centers and research facilities, and I was struck by when I walked into the lobby. I see computational medicine. It's not a name that I've seen anywhere in the world, for that matter. Why do you call your center the Computational Medicine Center?
We want to emphasize the two aspects of what we do. One is the medical problems we try to solve, and the other
part I get. I mean, I get, but
right.
but the word computational, like, why that word?
Because for us, computers are a catalyst for actually solving medical problems. Everything we do is data-driven. We do experiments in the lab, but unlike the traditional way of doing science, which was hypothesis-driven, we start earlier. We start with the data, and we ask the very basic question: what does the data tell us about this particular setting, this particular context? We may like what the computer tells us, we may not like it, in the sense that, what if it's not in the books? Then you have to answer a very basic question. Am I doing something wrong with the computer? Or is the computer telling me something I didn't expect, which is correct?
The computer just doesn't magically tell you these things. Like, your team has put a lot of work into writing programs, writing analysis to tell you something. So there's that aspect of it as well. So I suspect that's why the word computational. Maybe talk about that level of work that you've put in.
Correct. The members of the center do create the programs that will basically mine the data for something that we...
These are born programs that are born here, that your team is writing to tell you.
Or programs that we borrow from other teams that do similar work. But remember, these programs are written by humans, and humans can make mistakes. So
I've heard that. Present company, obviously excluded.
The complication is that when you see something out of the ordinary emerge from a program, there is always the possibility that I, the program writer, made a mistake along the way. And that's why I'm seeing something out of the ordinary. And it's not nature that I'm looking at, but Isidore's mistake in programming. So there's always that possibility. So you need to go back and check the programs again and again and again. Eventually, you sort that aspect out, and you're presented with the output of the program. Some of it is already in the books. You expect that because that means you modeled things correctly. Some of it is new. And now the question is, you need to sort it out, you need to prioritize, and this is, again, where computers come in. So in many ways, computers have many functions for us. The first is they are a framework in which we develop our tools, the computational tools. They allow us to quickly go through tons and tons of data in order to find this one gem that is relevant for this disease. Once we have it, we don't usually have one gem. We have many. So we use the computers to prioritize because at the end of the day, we need to enter the lab, the students that you met today, the postdocs, everyone who works in the lab needs to know what the first experiment ought to be. And when you have a sea of possibilities, the computer will tell you, well, try this one first, or try this one second. So we use the computer to get us to what used to be a hypothesis, but this is a hypothesis that's data-derived.
It's not just basic computing. Like you're talking about magnitudes of data, but you're also talking about another dimension. You're talking about high-performance computing as well. So it's a little bit more, right?
It's a little bit more. It's not the typical computer you would find at home.
No, certainly.
It's a high-performance system. Remember, my trajectory as a scientist includes computer science work. I have been in computer science for a little over 30 years. And I was always attracted to high-performance systems.
The budding part, the entering of IBM's relationship with you as well, is that's our bread and butter, high-performance computing, data, bread and butter.
The love affair began earlier, before, yes. Computers were intriguing to begin with, and in 1985 I had the opportunity to program a parallel computer. Now, back then that meant 128 processors, and it was exciting. You could actually see the possibilities. As a graduate student, I managed to work on a connection machine, and I managed to work with 32,000 processors. Well, 32,000 is a big number, and I could actually see the possibilities. So when the opportunity presented itself to join IBM Research in 1992, it was an easy answer because I knew that I could have access to things, to systems, to high-performance computing that mattered for the kinds of things I was doing. I had been in data-driven science forever, basically, and the IBM systems would make it a daily activity for me.
It's funny you say data-driven. Data-driven is a term that we use to describe what we do right now. Correct. In IBM, because we see data, and you've heard CEOs describe data as the new oil of the economy, basically. But in your case, it's much more than that. I mean, because the vastness of data, the amount of computation that you have to do, is a dream for IBM, because we want to solve problems like with that. With our technologies, so you use the word love affair. It's accurate because we see the work that you're doing as a challenge, right? And we want to bring technology forward that actually gives you the results that you need, protects your data, gives you the data. Maybe you could talk a little bit about that.
Yes, and in fact, you're correct. There are two aspects to high-performance computing. One is the number crunching aspect.
Right. Raw power.
Yes, raw power. But at the end of the day, you need data to process, and you need somewhere to store the data. So the second very big component of the way we practice high-performance computing is data storage. For us, data is a currency. It's the way we start the next experiment, the way we publish the next paper. So from the very beginning, when we started the center, having the capability to store a lot of data, to access it very quickly from our high-performance computing system, the number crunching as part of that system, and the ability to back up all of that data was a critical design consideration.
Yeah, I mean, absolutely. I mean, you think about it, like we've talked about 14 years of looking at this. A lot of data, 14 years. There's examples of people losing that data, you know, and imagine that. I mean, so we've worked really hard with you in the capacity of let's protect it, you know, let's make it available, and then let's crunch it. And you see that as very important because that's a lifetime's worth of work, absolutely, which could evaporate instantly had you not thought of it that way.
Absolutely. When we were designing the system in 2010, the system that is powering all of the Computational Medicine Center's activity, all of these were key considerations. They were basically the very first things we thought about, because without having very stable infrastructure, everything else we discussed would not have been possible. Can you lose data? Absolutely. And we did. So we're not immune. Even though we know how to do these things, we're not immune. I think we discussed recently about a little over a year ago, an accident ended up creating a loss of a lot of data that we had generated in the center over the course of about 8 years. Dozens of people having generated all this data. And precisely because of having paid attention to the design of the system in 2010, we were able to recover quickly.
It's got to you got to throw about a swing of emotion. You know, you lose all this data; it's invaluable. You can't put a price tag on, and then, bam, you save the day.
Yes, of course. Until we actually recovered the data, we went through some very trying times.
There's that gap.
Yes, there was a gap, but we were using the IBM Spectrum Protect solution for backups, and we were able to recover everything. It didn't happen overnight, but we did recover everything.
I mean, that's a good story. I mean, you know, this industry is not. You know, there's a lot going on with like malware, with ransomware, and there's examples of like years worth of research literally being lost and not recovered. So your experience is a good story. There, for every story like that is good. There's stories where data has been lost, and you can't recover from that. I mean, you literally can't.
We're glad we did it as well. Imagine these students that you mentioned before who are proud about their work. Their work is based on data. If the data on which they based the last 2 years of their thesis was gone, how do you replace that?
You don't.
Just exactly.
Yeah, it's a bad feeling. So I've really enjoyed the day. Clearly, you've got to be proud of your work, but it's evident that it's not just your work. I mean, talk to me about your staff.
It's not about my work. It's about the people who are behind the work. And it's the people you met today. Students, postdocs, lab technicians, my faculty colleagues. Without them, there would not be a center. And without them, we wouldn't be having a conversation about the new and exciting things that are coming out. And even though we've been discussing for a fair amount of time, we only scratched the surface of all of the exciting things that these people are actually producing. So in a very short amount of time, you'll be hearing even more. I come to the office every morning, and I'm excited to come because I know that they are excited to be here. I never had to tell anyone, oh, please spend another hour. do this because it's needed tomorrow. They know it. I don't need to justify anything to any of these people because they get it. The reality is that even though I'm sitting in this chair, without the people you met today, I wouldn't be sitting in this chair.
Congratulations for the work you're doing. It's tremendous work, and I believe you'll achieve your objectives.
Thank you. I'm very proud of them, and I can't say enough about what they're contributing to the bigger effort we're doing.
Thanks so much.
Thank you.
It was a real pleasure.
Likewise.